So good afternoon, everyone. Today, Ajiri, Anna, Chloe, and I will be presenting on mucopolysaccharidosis type 3. It's also known as Sanfilippo syndrome. So for our overview, we're going to be going over what is mucopolysaccharidosis type 3, the demographics, the etiology, the pathogenic cascade, signs and symptoms, diagnosing MPS 3, some examples of misdiagnosed based on neuropsych symptoms, the genetic perspective, the gene perspective, the molecular perspective, and the pharmacology perspective. So what exactly is mucopolysaccharidosis type 3? It is a rare genetic metabolic disorder, and it does cause fatal brain damage leading to dementia. And there are 4 different subtypes. So there's A, B, C, and D. And then in our bodies, we typically make glycosaminoglycans, also known as GAGs. And through our metabolism, it breaks down. However, in Sanfilippo or MPS type 3, there's a lack of that enzyme that normally breaks down the heparan sulfate, which is a type of GAG. As for demographics, because it is a rare disease, only about 0.28 and 4.1 cases per 100,000, and the most prevalent is MPS type 3A, and the most common subtype followed by that is MPS 3B. As I mentioned earlier, there is a lack of enzymes. So in each of these subtypes, these are the enzymes that are lacking. So in MPS 3A, it's the heparan N sulfatase enzyme that's lacking. And MPS 3B, the enzyme lacking is the alpha N acetylglucosaminidase. And then MPS 3C is the heparan alpha glucosamine N acetyltransferase. And then for MPS 3D, it's going to be N-acetylglucosamine 6 sulfatase. And then Anna is going to actually elaborate more on this later in the presentation. So for the pathogenic cascade, it's going to affect multiple cascades. And the primary storage of GAGs is the secondary disrupted pathway. So we're going to actually look at this diagram right here. So when we have the gene mutation, it's going to cause the deficiency of the lysosomal enzyme, which is going to lead to the excess storage of the GAGs. And then this could cause the dysregulation of the signaling pathways and the activation of inflammation. This is also going to lead to the impaired vesicle trafficking and the impaired autophagy, which is going to lead to oxidative stress. And as we all know, oxidative stress is not good for our bodies. And then it can lead to the mitochondrial dysfunction and then the calcium homeostasis and signaling. For signs and symptoms, early signs include speech and other developmental delays, getting many ear infections or sinus infections, large head size, respiratory infections, ongoing diarrhea, headaches, sleep problems, behavior issues that can look like those caused by autism, compulsive behaviors and hyperactivity. Sorry, you guys are in the way. Okay. And then for worse symptoms, seizures, severe cognitive problems, and progressive loss of motor skills. So for example, walking, speaking, feeding. And this picture of this girl, I actually found out about her through TikTok, and her name is Hayden, and she suffers from Sanfilippo disorder. I believe hers is type A. And her mom actually talks about her journey with Sanfilippo. So she started off with normal motor skills and normal developmental skills. And then as she progressed, I believe she's at 7 years old, her developmental skills are slowly degenerating. So for example, she can't walk as well anymore. She has seizures, she has trouble sleeping. I believe she can't sleep for like 3 days at a time sometimes. So if you would like to know more about Hayden, I can send you the information.
So diagnosing MPS 3. MPS 3, there's a few different ways that we can diagnose it. One is, first, it can be measured in the urine just because we can actually see how much heparan sulfate is being released. So we measure the enzyme activity through that. And so if there's a positive sample for this, that means there's increased heparan sulfate in the urine, which leads us to understand that there's a decrease in the activity of any of those 4 enzymes that Mariam was explaining. Now, genetic testing also allows the identification of the exact changes in the DNA, so we're able to see which specific type they have and then be able to treat them accordingly. So if the genetic diagnosis is known, it can be used for prenatal testing in the future, for future pregnancies, and also pre-implantation diagnosis. We can go to the next one. Awesome. So here's the autosomal recessive inheritance of the Sanfilippo syndrome. So just like they were talking about in the last presentation with Huntington's disorder, both the father and mother, they can be carriers and have that defective allele, but have no idea that they're passing it on to their children. And so we can see with this case, both the father and mother are heterozygous for the affected allele, and one of their child will have both alleles and be affected with the actual Sanfilippo syndrome, while the other three will be unaffected. Two of them will be carriers. You can go to the next one. So from the genetics perspective, just like Mariam was talking about, each one of these different types is caused by mutations in the genes. So MPS 3 type A is caused by mutations in the SGSH gene. MPS 3 type B is caused by the mutation in the NAGLU gene. MPS 3 type C is the result of mutations in the HGS-NAT gene. And then for type D, it's caused by mutations in the GNS gene. And these genes are going to code for enzymes that Anna is going to talk about a little more in the breakdown of heparin sulfate. You can go ahead and change to the next slide. So even aside from the different genes, so I was actually able to look online, and unfortunately we already turned in our final presentation, but I was able to find out kind of the different breakdown of mutations and what was caused. And basically with the type A, because it's the most aggressive one, it had actually most of the mutations in the actual gene sequence, where type B, it was around the same, and then type C and D didn't have as many mutations in the gene. So even aside from that, Sanfilippo type A, there have been examples of misdiagnosis just because of what Mariam was saying. A lot of times these children are born and they're able to walk fine. Even in this case, there was a man that wasn't diagnosed with this disease until he was 28, which is really unheard of. Because when he came out, he was fine. He started walking at 14 months, just like a normal baby. He, he may have had some speech delays a little bit, but for the most part, he was doing okay as a kid. He was able to learn how to ride a bike when he was 12. And there were only just a few non-specific neuropsychiatric symptoms. And so they really weren't sure what it was. He had some mental and speech development delays, but they didn't do any urinary testing. Age 12, he had a little bit of regression in motor skills, speech, cognitive abilities. Then around age 14 and through age 18 is where he started to really have issues where there was that restlessness that Mariam was talking about, excessive involuntary movements, repetitive movements or sounds. At 16 and 18, he actually lost the ability to walk and speak, and they still couldn't figure out what it was. Age 24, he had severe hearing and visual impairment. And then finally, the parents were able to bring him to a neuro center, and they were able to do an actual genetic testing and figure out that the entire time he had Sanfilippo. And normally for Sanfilippo type A, actually patients only live between the ages of 15 to 18. So the fact that he even was still alive by age 28 was like a medical marvel in itself. And so then they were able to kind of give him some sort of treatment to alleviate some of the symptoms he was feeling. But they have no idea just because it can be easily missed. Go ahead, you can change to the next slide.
So I'm going to discuss more of the molecular perspective of mucopolysaccharidosis, starting with this picture of an electronic microscopic view of a cell. Here's the lysosome, where the arrow is pointing. Yeah, there, right there. And this is the organelle that is going to be involved in this disease. So as we can see for the name, mucopolysaccharidosis is an excess of mucopolysaccharides. And these molecules are large polysaccharides that are found in mucosa. So for example,
in synovial fluid, that we find some saccharides, which are in this classification, same as hyaluronic acid, for example, is in this classification too, but in this case, it refers to the mucopolysaccharides that are building up in the lysosome. So this diagnosis is established by the pathogenic enzymes that come from those defective genes that are mutated. Next. So here's a picture of a study, an animal study in the cerebral cortex of mice. You can see in the wild type, the arrows are pointing at a lysosome, and it looks very different than in the MPS 3A lysosome. As you can see, the ones at the bottom look more diffuse because the lysosome is being more burdened by that buildup of the mucopolysaccharides, or the GAGs, which means glycosaminoglycans. Is just another broad name for it. Next. So as we're discussing, these enzymes have different functions, but the goal of all of them is to work together to break down this lysosomal GAG. So in type A, the enzyme affected is N-sulfoglucosamine sulfohydrolase. Its function is lysosomal degradation of heparan sulfate. This is in the category of sulfatases or sulfohydrolases, and its function is to break down or to like hydrolyze sulfate group out of the saccharides. Type B, we have alpha N acetylglucosaminidase. This is a type of acetyltransferase, and its function is to break down large glycosaminoglycans. Next. Type C is heparan alpha glucosaminidine and acetyltransferase. So this is another different acetyltransferase, and it degrades the lysosomal heparan sulfate too. And type D is N-acetylglucosamine 6 sulfatase, another sulfatase that takes away the sulfate group. Its function is catabolism of heparan sulfate, and keratan sulfate. So the three of them have a sulfate group, and they're considered a type of glycosaminoglycan in the lysosome. Next. So here I have a picture of the actual molecule. This is a part of it. It's a long chain of these repeating, they're called monosaccharides, or uronic monosaccharides, and sulfate monosaccharides that are repeating in a chain. As you can see, the sulfate group and also the carboxylic acid group that is in the sulfates, that is in this saccharide. So the enzymes have to first take away the sulfate group and then take away the carboxylic group, and then break down this large molecule. So they have to work together in steps. So if one is missing, then that step cannot be completed and the molecule will build up in the lysosome. This one in particular in the picture is heparan sulfate. It's a proteoglycan, and it aids in cell signaling, angiogenesis, and coagulation. So for example, it is present in mast cells, so it has a role in the immune system. Lysosomal accumulation of heparan sulfate in microglial cells, followed by their activation and cytokine release, result in mitochondrial dysfunction in the neurons, causing their death. So the accumulation of these molecules will ultimately result in the death of these neurons and in brain impairment. Next.
Okay, so I'll be talking about the pharmacology perspective. So for now, there is no approved treatment for MPS 3, but that has been shown to be effective through certain therapies, such as gene therapy, enzyme replacement therapy, known as ERT, and substrate reduction therapy, known as SRT. So in enzyme replacing therapy, it involves replacing the enzyme that is missing in the Sanfilippo enzyme produced in the laboratory. And there are some difficulties with using ERT, and such as crossing the biological, the blood-brain barrier, reduction in the cost of accessibility of therapy, delivery to difficulty sites of the pathology, early diagnosis of the treatment and immune response to ERT, but there have been some positive outcomes where people have been found to have more joint mobility, walking ability, and respiratory, and less respiratory infections. But with SRT, it's more found to be used because of the issues, the challenges of using the enzyme replacement therapy. The next one. So with substrate reduction therapy, small molecules are attractive as a therapeutic agent for MPS III because they have the following advantages. So basically, SRT is known to reduce or decrease the toxic accumulation, and it's been found to be more used, like I said before, because it's easier and it's able to pass through the blood-brain barrier as enzyme replacement therapy isn't. And so low cost, there's also stability, it's able to cross the blood-brain barrier, it's non-immunogenic controlled dosing, and it has multiple routes of administration. And then there's some alternatives. So in MPS III, neurodegeneration is the main debilitating clinical signal requiring efficient CNS targeting. And so in vivo gene therapy and ex vivo gene therapy, and then also enzyme replacement therapy as well. And with gene therapy, like the last group said before, the most used virus to basically gene therapy, you're using the gene to replace. It's almost like an instruction to get them to produce the enzyme for Sanfilippo. And the most common used virus is the adeno-associated virus. And over 8.5% of normal enzyme activity will be sufficient to improve the neuropathology and the behavior of MPS IIIA seen in the mice models that were treated with gene therapy.
Chart, you can see kind of what Ajiri was explaining before about how if you have Sanfilippo, you see that there was a change in one of these genes here, and it led to the deficiency. So all of this heparan sulfate is going to accumulate. But with the gene therapy, if we're actually delivering a functional copy of that gene into the body, it provides the instructions that the body needs to make that deficient enzyme so that that heparan sulfate is degraded like it's supposed to be. Go to the next slide. And so here you can see there's different ways that for each different type, there's different ways that this gene therapy has been delivered. Like Ajiri was saying, there's multiple routes. So for type A, they're using viral delivery vehicle. Type B is a stem cell based gene therapy. For type C, they're using like what they're using in Huntington's, the AAV9 based gene therapy. And then for type D, currently they have not started clinical trials for a gene therapy just because it's such a rare subtype of this disease. And so they haven't decided how they're going to kind of attack it treatment-wise yet.
So in conclusion, Sanfilippo syndrome is a debilitating condition that typically results in shorter life expectancies. And though there is no treatment, there is no cure for it, there have been drugs and therapies shown, like the ones I mentioned before, that could arrive to the cure.
Thank you so much for listening to our presentation.